At FluoGuide, we maximize surgical outcome, and we do that by. So may I have the slide, please? Is that? Thank you very much. So just a forward-looking statement, and so you have to make your own assessment. So if I have the next slide, yes, thank you. So really what we do at FluoGuide, we maximize surgical outcome, and we do that by intelligent targeting. We start out by guiding cancer surgery, and that's because there's a huge unmet medical need. So there'll be about 15 million patients every year that have cancer, and about 80% of them need surgery, sometimes during their disease. And half of the cases where the surgery is done, cancer is left behind. That's not because it's because it's very difficult to see for the surgeon. It's very hard to see where the cancer stops and where the normal tissue starts. And our solution to it is very simple. We have a UPAR targeted illumination of the cancer, so the surgeon can remove all cancer the first time. We already now have a first product in clinical development and shown that it's safe and that it lights up human cancer. So a very good foundation. We have a strong IP protection, strong cash position. We have a market cap about 1 billion SEK listed on the First North in Stockholm. And then we have a cross-functional team that is prepared to execute. May I have the next slide, please? Our procedure is a very simple one. We inject our product prior to surgery. It will circulate in the body, it will bind to the cancer, and then during surgery, the surgeon can switch on the fluorescent light, and they can see precisely where the cancer is. So one of the things that is very interesting with FluoGuide is that everybody likes us. I mean, patient obviously because we help them to be better cured for their cancer. The hospital can potentially save money on less operation, less aggressive treatment. The surgeon become better to do what they really would like to do, namely treating the patients. And last but not least, equipment manufacturers, we get more features out of or proof and we're going to say enhance the use of the features for the equipment of the equipment manufacturers. So they are also quite pleased with us. So if I have the next one. So really what we do is that we are targeting an enzyme system that is sitting on the forefront of the cancer. And that's sitting on the cancer cell and is used by the cancer to tear down normal tissue, so it's able to spread into the normal tissue. And it means that it's the more malignant the cancer is, the more UPAR is expressed. And also means that the UPAR is expressed particularly on the forefront of the cancer, and that's exactly where it's difficult for the surgeon to see it. I mean, they can easily see the normal tissue, they can easily see the bulk of the tumor, but really what's hard to see is where the two things are moving into the other. So we have based our technology on a long decades of work of research. It's been used, UPAR been used for drug targeting, diagnostic purposes, but we're the first one taking it into using it for guiding surgery. And we have done a series of in vivo studies to prove that it works, and that is being done by human cancer, incorporated into to animals, and then we've done the surgery on these animals. And as you can see on the right side of the slide, you can see that the delineation between the tumor and the healthy tissue is very precisely marked by our product, and it's very aligned with the anatomical difference between the two of them. So if I may have the next slide. One of our studies, just as an example, is where we've taken a human surgery with human equipment with a human cancer. And then we asked the surgeon to remove all cancer under white light. They did so, and in the 4 of the animals, there was 8 in total. In 4 of the animals, they removed more cancer. Then we switched on the fluorescent light, and the surgeon could then see our product. They could not see it under white light, so they could see it then when we switched on the fluorescent light. And then 4 more of the animals had cancer removed. And had it been patient, they have been sent home. They would have been coming back to the system. Either they have had relapse of the disease, or they had poor quality of life, or maybe ultimately they could have died from the disease. So this is really how we help patients and society. I think one of the things that's very important is that we have used standard equipment. We don't want to go out selling equipment. We want to use equipment that is already available at the surgical place, and that's why we use the standard equipment. Also, if the standard equipment is not necessarily the best of the equipment, we still want to be able to work and deliver our benefit on those equipments. If I may have the next slide. So we're targeting a market about 5 billion euro for our near-term plan, and that is to go after 3 of the prevalent indication as well as glioblastoma. And the reason for that is that, why we have selected those indications is that UPAR is expressed on, extensively expressed on most of the solid phase tumors, and that is including lung cancer, breast cancer, and colorectal cancer. It's not so much expressed on the prostate cancer because prostate is generally not so aggressive, but for the 3 others I just mentioned, it's extensively expressed. We don't want to have a screening program when the patient comes in. We just want to be sure that the surgeon knows that it will work on the patient, and they can include the patient into the treatment, and that's it. Some of the other indications, like the high-grade glioma or glioblastoma, which we are focusing on first, is more rare, but they have, on the other hand, an orphan drug designation potential. And that is one of the reasons why we have started out in glioblastoma. So if I may have the next slide, please. So glioblastoma is a very severe indication. There's about 1 out of 20 patients that will be alive only in 5 years. And that's because glioblastoma is so invasive into the cancer. It's very important that you can see, if you should remove it, you can see into the tissue because it really invade deep into the tissue. Another point is that, as it is the brain, it's very important that you remove the cancer very precisely. You cannot just remove everything, because then you would also remove some of the more critical functions such as speech or movement, and that of course is not possible, because then you will give the patient more disability than you can give them benefit for removing the cancer. So neurosurgeon is a very difficult dilemma when they do the surgery. They really have to choose between removing all the cancer and do it precisely. One reason as well why we have chosen this indication is because the neurosurgeon already now are well trained and skilled in doing the surgery. They have a lot of equipment there and it's present at the theater. So it's really a plug-and-play situation. And if I may have the next slide, please. So one other thing as well that is very interesting with FluoGuide is that we have a very short and direct way to the market. That's because our studies are small and we need few patients. And the reason for that is that our endpoint is a positive predictive value. And what that means is that when the surgeon removes tissue that lights up and then after is validated by a pathologist that is in fact cancer, then we have a positive predictive value of our product. And that's the endpoint we need in a phase 3 study to get our product approved. Importantly, this is done in the same way as I just shown before with the animals, that the patients are included, the surgery is done under white light, the fluorescent light is switched on, and is validated by pathologists that the surgeon removed just the cancer and not more. And that means that we will need about 150 patients, like for proving efficacy, and then maybe need some more, maybe 500 for proving safety, but that can be pooled across different indications. And before starting that pivotal study, we need to do a phase 1/2 study where we establish the safety, find the optimal dose, and that's the study we have ongoing right now. Additionally, for that study, what we also get out of it is that we get a proof of concept and we get an understanding of the benefit we can add to patients, and therefore the price we potentially can charge for our product, which of course will be very important for the value of FluoGuide in the end of the day. So this is very much the program. We are under the drug regulation, but we are an imaging agent. So if I have the next slide, please. So the study we have ongoing right now is the first one of the 2 I mentioned. So it's a phase 1/2 study, and it's divided into 2 parts. And we have now treated 5 cohorts in the first part of it, and each cohort consists of 3 patients, and we treat 3 patients on a given dose. And when it's proven safety, we increase the dose until the next dose level, and then we keep going. And as you can see, the 16 is not adding up. It should have been 15, but that was because an accidental additional patient was included in one of them with another cancer. I come back to that in a second. But we are now seeing that it actually lights up and that it's well tolerated. And that's really, really nice in the sense that we see it as a flaw in our case. Now the question is, how good can we make it? How much benefit can we add to the patient? How high a price can we get? So we expect both the remaining part of the part 1 expect that in Q3. And then we will go into the second part of it, which basically is exactly a mini phase 3 study, just with fewer patients. So we do the same equipment, same surgeons, same endpoint, same design, same anything, everything. It just that it's fewer patients. And that's really to train us to do the phase 3, and that's to have good quality end of phase 2 meetings with FDA and Inga to be sure that we get the pivotal study right. So as I mentioned, one of the patients was included with a non-brain-derived cancer, which was a very, it often happens because it can be difficult for the neurosurgeon to see if it's glioblastoma or another cancer. And sometimes they do have patients that are included into the surgery that do not have the glioblastoma. For us, it was quite important because it was a non-brain-derived cancer, and actually give us the first hint in humans that it also worked outside of the brain. We did not expect any tolerability issues because we had done a very extensively done preclinical package, and we did a 4-week study. That's enough for us to get approval, and we were not limited by toxicity but by feasibility. So that means we couldn't get more product into the animals before it was limited. We had to stop, and that's a dose that is more than 100 times higher than the one we expect in humans, and that means that we really have a huge room up to the maximum dose we can give and up to what we expect will be given any probability. So if I get the next slide, please. So I think that our pipeline now is that we have the first indication is glioblastoma for our first product. Then we will start a prevalent indication study after we have the first part study data, and that means when we have the safety data, that really when we get out of the very constrained phase 1 setting, and then where we can include more patients quicker, we don't have to treat them one at a time, we can treat more at a time. And that really is what we will do after summer when we have closed the first part of the study here. It would be lung or breast cancer that would be selected as a prevalent indication, and we promise one prevalent indication does not prevent us from doing more. And we have very strong interest for the community to do more, but we will take it one at a time. Then we have a second product that we licensed in last year. It's FD2, and it has another profile than FD1 in the sense that it's excreted in the kidney, and that means that it's better suited for stomach cancer, where FD1 is excreted in the liver and better suited for upper body cancer such as brain and lung and breast. So we had another interesting addition to our pipeline, that was the photothermal therapy that we included or licensed in earlier this year. And that's really an effect we see with FD1, that we can heat up the product in the sense that we kill the cells that FD1 is bound to. And that means that we can kill the cancer cells. Interestingly, the cells where there's no FD1 bound to will not be heated up by the light in the same way, and that's just getting a temperature just below the tissue damage. And that means that very precisely deep into the tissue, both can kill all cancer cells, and at the same time we can spare the normal tissue. And if you remember the situation before with the center of speech, the neurosurgeon had this dilemma if they should remove it or not. Now they can actually leave the center of speech, but they can really sweep it with light, so they kill all the cancer cells invaded into the center of speech, and that way it both destroy and remove the cancer, but at the same time preserve the function, which is a very, of course, important benefit that we expect will be appreciated and therefore also paid a high price for. Yes, if I get the next slide, please. This is an example of how the photothermal therapy will work in practice. We will still use it for guiding surgery, and when the surgeon is finished, they can just add on the photothermal therapy on top of it, and then they can remove any leftover cancer or cancer invaded into critical structure in the brain. So if I may have the next one, please. This one is an overview of the competitive landscape. Two products was approved about 15 years ago, or roughly, for the first time. The first one was Hexvix from Photocure, and the other one was Gliolan from a Japanese-German company. They are special in the sense that the product that light up is not, or have a fluorescent property, is not the product given themselves. It's actually a metabolite, and that means they form a kind of a cloud around the cancer. They're not bound specifically to the cancer, but is converted to the fluorescent molecule inside the cancer. It also means that they are limited to one indication each, depending on the administration. So for Hexvix it's bladder cancer, and for gliolan it's glioblastoma. And last but not least, they are using visible light, and that means you can only see it 1 to 2 millimeters into the tissue. Basically, you can only see it on the surface of the brain. So, for instance, when you look at the brain, you can see if the cancer is getting out to the surface. But if you have the near-infrared light, which all the rest of the product are using, then you are able to get 1 to 2 centimeters into the tissue. And there's none of the new second generation product that is approved yet. They are all based on this near-infrared light where they can see 1 to 2 centimeters into the tissue. They're all bound to a specific receptor or protein or molecule in the body. Some of them are equipment dependent. That's one of the things that we would like to stay away from. We would like to be equipment independent in a way that we can be used for all existing equipments. And I'll mention just 2 products. One of them is SurgeonMap in phase 3 for colorectal cancer, and the other one is on target in registration phase with ovarian cancer. And I think that what is really unique with FluoGuide is that we have the near-infrared light, we can see deep into the tissue, we're equipment independent, and we are very cancer specific on the one side, but cancer type unspecific on the other side, and which really means that we can go in for the glioblastoma and then we can spread out to all the other more prevalent cancers from there. I think also it would be naive to believe that any of us will kind of dominate the entire market. We have to divide it between us, but I still think this is good because this is a very huge market, and actually by having more actors working on the market, we're able to develop it. And I think that's what Photocure and this gliolan product have started to do years ago, and now there's really come and quite a lot of interest into it. If I may have the next slide. Some of this interest is coming from the evolution taking place in the surgical field, and I think this is the second thing that is really, really important for FluoGuide. That is that we are riding on some trends in the surgical improvements or quality that really have gone up, of course, for some decades, but still is going on. And that with the equipment coming into the surgical scene means that more benefit can come out of the features from the equipment manufacturers, and the surgeon become better to do what they want to do. Some of the most penetrated market with equipment is neurosurgeon, and they are penetrated with microscope because it used that even before the fluorescent guided surgery or came up. And every neurosurgeon are using a microscope. Then on other indications are less penetrated, but some of using the scopes very much, stomach cancers, gynecological cancers, and also lung cancers. And then now starting coming the robotic surgery that goes into many different indications. But interesting, and what is important for all of them is that really the drivers of this development, the digitalization, the minimization, and automation is really helped by the fluorescent guided fluorescent guided development companies are really helping the equipment manufacturers to get the most benefit out of their equipment, and that's really where there's a strong synergy between all of us, and that is really a strong trend that is that's going on. All of the companies listed up here is some we are talking with. I think in all fairness, we will not have a monetary deal coming out of the brain cancer. It's a quite well-defined market, very concentrated. But I think it's likely that it will come out of in a more prevalent indication such as the lung, the breast, the colorectal cancer, where many of the larger players are really putting a lot of attention into because they are larger markets. If I may have the next slide, please. So the team behind, as I mentioned, we are cross-functional. We start out with Andreas Kjær. He's board-certified nuclear medicine chief physician at Rigshospitalet in Copenhagen, and then he has a professorship at the Copenhagen University. He has a group about 50 people working on imaging, diagnostic, and UPAR cancer, and that's where we kind of was spawned out of. It also means that we have no research department. We actually license and acquire products coming out of university, so we don't have any fixed cost, but we can take what is coming out of this very very interesting group. The next one in line is Grete, taking it through production and manufacturing and preclinical testing, then coming to Detta, the clinical testing. Henrik, we just was joining the company just end of last year, and it was because we're becoming more commercial now. Henrik has been in the field for a long time and a CFO in life science company, and was actually part of the very first one in Denmark many years ago back. And he was very instrumental in the directed issue we have just completed. Myself, I've done companies before, and I have done Nanovi before, and that's an oncology company taking the invention from the bench to the market, and that's also what I want to do now with FluoGuide. So if I may have the next one, please. We very recently, a few weeks ago, we closed a directed issue, a very important one for us. It was 75 million SEK, but the important part of it was really that it was very good names, institutional investors, and we got about 15 new investors into the company. Importantly, also many of them came in with an intent to test us out, and assuming we deliver what we promised and what we've done before, then they would like to invest more. So really what this does to us is that our shareholders giving a lot of robustness and give us flexibility in the partnering versus financing later on, and also it gives validation basically to the company. We was we was IPO'd in 19, and we have had a very good development since that. It was awarded as the best IPO in 19, and we also have had some very good funding of EU. We got 2.5 million euro funding, and we have had a very incentivized team. We also have approximately 10,000 shareholders in the company. So if I have the next slide, please. So to sum it up, I think there's three things that is very important with FluoGuide. Excuse me. The first thing is that we have a very big market, 15 million patients every year with cancer, and about half of them, or 80% of them, need surgery, and half of them recur after surgery. We have an important technology to be able to help UPAR elimination of the cancer. We have very important trends in the market: digitalization, minimization, and automation, and really that the fuel the development of FluoGuide and the field. And then last, we have a product that now has demonstrated to be safe and well tolerated, and really we have very good way to get the best out of this market. We have some important milestones coming up: safety Q3, proof of concept in Q4, sorry, for our key product and glioblastoma, and then we will start the prevalent indication after summer. Also for the next year, we have some very transformative milestones. We will be starting a phase 3 study in glioblastoma and with FD1, and that really transforms us into a late-stage clinical company. We do start the photothermal therapy development, giving higher benefit, higher price, higher value to the company, and then of course we will also start our second product, give us more flexibility in the price differentiation and partnering. So thank you very much for your attention. I'm open to questions.
Yeah, thank you very much. Maybe one question on the regulatory pathway for this drug. How would that look like?
Yes, I mean, we are under the drug regulation, so basically we follow this phase 1, 2, 3 studies, but under that there's an imaging guideline, and that is what we also follow. That means that it's more device-like in the sense that the phase 1 and 2 is put together, and the phase 2 and 3 is also put together, and that's why we only need 2 clinical studies to get approval, and we will get the registration in 24 for our first product, first indication. So it is a drug regulation, but very much a device philosophy.
Okay, yeah. Well, thank you very much for your presentation.
Thank you very much.
Yeah.